Log on, tune in, find out. Another good idea from Cambridge.
We're very pleased to welcome Professor Andrew Wyllie, who will be known to many of us here, who's Professor of Pathology, is head of the Department of Pathology, who's been there as long as I can remember, actually. How many years was it? I don't remember. Anyway, it's been there for many years. And of course, Professor Wyllie is particularly well known for his research in the area of programmed cell death, the regulation of cell life and death, and so forth. And indeed was there at a very early stage during the early discoveries that were being made on apoptosis and on this whole area. So we're very pleased that he's come to speak to us on a very busy schedule, and his title today is How and Why Cells Die, Biological and Theological Perspectives. Thanks very much, Professor Wyllie.
Well, thank you, Dennis. Thank you for the invitation, and thank you for coming. I have been warned not to go on and on and on. And actually, for your reassurance, I think the battery of my computer, which is powering the talk, may well let us know when I ought to finish. Hopefully well before 2 o'clock. So how and why cells die? Of course, how is an easy question in biology, why much more difficult. And so we're going to try and mix these together, and I'm going to cheat a little bit, because as you'll notice halfway through the talk, I'm going to change gear, I think, and answer or attempt to answer another but related question relating to why. So, let's see if we can. It works, and this works as well. What you're looking at is a phenomenon of cell death under entirely physiological conditions. What you're seeing is a little fragment in scanning electron microscope image of the connective tissue, the fibroblast generating tissue, in an area of a chicken wing, which is built and then unbuilt again in the formation of a wing. Very similar things could be shown in the development of the mammalian hand, which does not develop as a little knob, which is the palm, out of which sprout 5 fingers, or 4 fingers and a thumb, but it develops as a paddle-shaped structure, and then the fingers are shaped by a process of death of the web that connects them. So cell death in development is a very common event. You can see here these rounded cells, this one here. I always think they look a little bit like an old-fashioned sponge. And it might be that this audience is more attuned to what I mean than some of the audiences, the student audiences that I speak to. But you can see this extraordinary, the cell has lost this sort of laminated structure where the fibroblasts are stretching out to touch one another. And what you see is a rounded cell body, it's losing contact with its neighbours, and it's got these little tantalising little holes in its surface which seem to lead into a mysterious interior, which we see a little bit of here. This is a section actually through a dying cell, and somewhere down in here you can just see the outline of a living cell, and you can see the big nucleus with the chromatin finely fenestrated to the pattern there. But you can see that this nucleus looks very different, and what's happening is the chromatin, that's where the DNA and the protein are that really dictate what sort of cell this is going to be. This chromatin is condensing underneath the nuclear membrane. This funny looking object here is what remains of a somewhat altered nucleus, and then there are these odd sort of vacuolar structures inside the cytoplasm. If you were to look at these in more detail, many of them are actually normal cytoplasmic organelles, but packed together more closely than they would normally be in a cell whose lifespan is not limited. This one is very near finally giving up the ghost. And you can see here what happens around about the same time, the cell, I'll show you this in video just in a minute if it works, but the cell throws out blebs and blisters, which it then withdraws and throws them out and withdraws them. The whole surface starts to boil as the cell realizes it's coming towards the end of its natural lifespan, for whatever reason. And then eventually breaks up into a series of membrane-bounded bodies. And these bodies, they contain little bits of cytoplasm, still recognizable. They will still exclude the sorts of colouring dyes that biologists use to identify living cell membranes. So they've got a lot of life in them still. But on their surface, they are expressing glycoproteins which normally are hidden inside the inner lamina of the cell surface. These are appearing on the outer lamina, and these are signals for their neighbours to come and ingest them. And this is what actually this picture is all about. You can see this is the dying cell, this bit here. There's its nucleus, now heavily condensed, little bits of organelles here and there, but this cell has come up and is embracing the dead cell. Can you see that thin sheet of cytoplasm surrounding it? Of course, this is a section, so what you're looking at is the living embracing and about to engulf and digest the dead. Cells are a nasty piece of work when it comes to organising their society. But you can see how this type of death affecting single cells, and, oh, by the way, here's a three-dimensional image similar to what you saw before in the chicken wing. This is actually a thymocyte, not recognizable. People that are used to looking at thymocytes like Dennis know that they look a little bit like sea urchins with lots of little finger-like processes that wave around. And here, that's all disappeared from the surface, but you see again these interesting little cavities which lead into these distended portions of part of the cell we call the endoplasmic reticulum. So, this is the morphology of cell death of the sort that happens naturally. Now, the interesting thing is that it happens naturally in development in order to shape organs, but it also happens when cells undergo periods of stress. So even the pathological death of cells that have been stressed, it recruits this mechanism, which is a mechanism straight out of normal physiology. I always hesitate. The good cell biologists always have wonderful pictures that you know, at a flick of the wrist, they can make to work. I have a moment of terrible anxiety as I approach this picture, because, right, what you're looking at is a monolayer of cells growing in the bottom of the dish, and when I start the frame, you'll start to see the cells all jigging about. This is quite normal. They move around a little bit and they touch each other and shoulder around each other, like people standing at a bus stop or in a football queue. And what you'll see is a gradual enrichment in cells which are coloured this pale green. The dying cell goes through a period when it picks up this pale green colour and allows us to recognise them as they die. So now we'll see if we can start. So it's jigging around. You may find it useful to sort of look in this area here, and I think you'll see, yes, cell just pops up over here now. See? And they look like little bunches of grapes at this stage. The cell loses its morphology and goes into these little clusters of small bodies, and gradually the field is filling up with these dying cells. The speed is, of course, you know, the hours are ticking past, as you see. But it is a process with very distinct morphological characteristics. Now, the next thing is to get it to move on. It would be rather tragic if I could not move. I don't want you. How do I get you? So, are there any other characteristics of this type of death which would allow one to say this is something discrete. It's not just a cell falling apart. It's not falling off the edge of its lifespan. It's actually doing something in order to die. And as it happened, one of the entry points that we had to this story was we were able to purify these dying cells with their condensed nuclei from their neighbours. And this is actually death induced by a physiological hormone in vitro, and sometimes the cells die and sometimes they don't die, not immediately. So these two came from the same steroid-treated dish. This guy was in a similar dish but no steroids, so he's our absolute control for a living cell. And what happened here was that we extracted chromatin from these cells' nuclei, took the DNA out of that chromatin and ran it on this agarose gel. And what you see is this bright signal here indicating very high molecular weight DNA in the DNA of this nucleus, which is what you would expect, because there's about 1.5 meters of DNA packed into each nucleus in something of the order of 40 chromosomes. Now, the difference was when we looked at this nucleus, and you can see here the DNA is coming down into the gel, indicating it has a component with very low molecular weight, and then the increases in molecular weight have a staged step-like structure. This actually tells us something quite fundamental about what's happening inside the dying cell's nucleus, because nuclei are arranged so that their DNA is wound around little protein spools called nucleosomes, and then the spools are then wound together, and then the spools wound together are wound into yet bigger structures, which eventually we call chromosomes. But to pack that 1.5 meters into a tiny nucleus is quite an exercise, and to find what you're looking for inside that nucleus is an even more remarkable process. But that's how nuclei work. And the point about this gel is that it demonstrated a characteristic change in the formation, in the structure of the chromatin. It had been cleaved in between these nucleosome particles, these little spools. And you can see that that's why it produces this easily discerned ladder. This is just 1 spool worth of nucleus, that's 2 spool worths, that's 3 spool worths, and so on as the length of the DNA increases. So I guess this was one of the first observations which said there is a biochemistry, there's a cell biology of cell death beyond morphology alone, which for some reason non-morphologists are very suspicious of morphology, so they want something biochemical. This is not quite biochemical, but it's getting on that way. It was a marker which for some years was important in identifying cell death, and there are still various kits that you can buy now to demonstrate the presence of this fragmented chromatin characteristic of the process. I was at that point a PhD student. I had a very inspirational mentor, a gentleman called Alistair Curry, who was the chairman of what was the Cancer Research Campaign at that time. And he was insistent that we needed a name for this process. And the name that came sort of out of the woodwork was apoptosis. Out of the woodwork? It actually came from a professor of classics in Aberdeen, Aberdeen being the sort of place where professors of classics and professors of pathology occasionally had lunch together. And Alistair said, could you give me a name which would describe a process whereby single cells die within living tissues? And he came up with apoptosis. Being a good Scot, he said, you don't pronounce the second P. Years later, after all the mockery of introducing a new term, particularly a new term with the letters P-O-P in series stuck in the middle of it, there was a lot of laughter. We appeared actually in that year's Lancet Christmas quiz as what is apoptosis. Yeah, it was all a bit real grievous. You're trying to do science as an aspirant PhD student, and what people do is, oh, that's a real joke, you know, how funny, you know, a funny word has appeared. Well, anyway, years later, you go to Greece, the system has changed. Everybody is studying cell death. You give a talk not terribly dissimilar to what you just heard, and the Greek professor approaches you at the end of the lecture he says, "You know we still use that word." What for, you ask? Just for a moment you think there must be some ghastly obscenity about it here. He said, "No," he said, it still means falling off. You know, we use it for priests that are dismissed. Well, when did it stop being a joke? There were one or two additional things of enormous significance. Here is a slide which represents, in just one slide, the enormous work from the laboratory of Bob Horvitz in MIT, studying an organism he was introduced to here in Cambridge in 1972, Caenorhabditis elegans, a nematode so small that you can identify with special microscopy every single cell in its body. There are 1,081 somatic cells in this adult organism. Moreover, the development is hardwired, so that you can predict exactly where in the worm a particular cell will divide and produce two, we say daughters, but in fact sometimes it's a son and a daughter. Son goes nowhere and the daughter goes on proliferating. And so you can build the whole lineage beautifully reconstructed. And so predict where a cell is going to die. And then it turns out, of course, this organism is very easy to mutagenize, and so you can begin to look at point mutations which regulate cell death. Bob Horvitz always likes to say at this point in the lecture, at this point I knew that there was a biology of cell death. So the question of how is well on the road now of being resolved. And what he showed through a whole series of genetic experiments was that there is a protease family, protein digesting family of enzymes, which activates the process of death. It cleaves about 200, 300 molecules of different sorts, but all of them interesting molecules that are key points in the cell's lifespan. It cleaves them and destroys their activity, or sometimes switches on activity as a result of the cleavage. It is an active process, and it is started off by upstream molecules which are related to the mitochondrion, which is the sort of energy supply of the cell. So here is death that's tapping into cellular energy, cascading or turning on a cascade of molecular events which diffuse through the cell and cause actually most of the morphological changes I've just discussed with you. The next remarkable event was when it turned out that exactly the same type of process occurs inside human cells. The human cell has another branch which links with the exterior instead of the interior mitochondrion, and up here a similar family of proteases are activated, turn on these effectors and push the cell down a pathway in which it effectively digests itself. Moreover, the proteins here, the protective proteins that stop the process and keep the mitochondrion stable, are directly homologous to the C. elegans proteins doing the same job. So this is a very ancient pathway. Not all metazoans have these pathways, but most do. So I want to show you next, just in the next 2 or 3 minutes, an experiment we did with a very notable lymphocyte biologist, Mel Greaves, who asked us for some of the material that had been developed in the laboratory in Edinburgh where we were working at the time. What we were looking for was the upstream regulators of cell death. And at that time, people had recently become very interested in a molecule which is called P53 because it has a 53,000 molecular weight. And it has attracted attention of the cancer world because P53 is one of the genes that is usually either mutated or deleted in most cancers. So the experiment which we were able to do, because there was a very bright young man working in the next lab to me, called Alan Clark, now professor in Cardiff, he was able to knock out P53 in the germline of animals. These animals then grew quite normally without P53, suggesting that maybe P53 wasn't necessary for normal development, but it is necessary, it turns out, for the reaction to certain types of DNA injury. So, what you see here is a depiction of the cells as they're moving around the cell cycle. Those of you who've seen this, forgive my Mickey Mouse explanation, but I'm conscious of time going past. What's happening here is that for thousands of cells at once passed into an instrument, one's measuring the amount of DNA on this axis within each cell and counting the number of cells with that amount of DNA on this axis. So it turns out that there are two major populations of cells in any growing cell culture. One, cells that haven't yet replicated, and they've got this amount of DNA, and one in which they have replicated and they've got double the amount of DNA. And then, of course, when they go through their cell division process, they slip back to having just the one baseline set of DNA molecules until they replicate again. This little peak here is what you always get in a cell culture. It's a small proportion of the cells that are dying in any case. Right, okay. So we take out these cells from the marrow of a normal animal and we irradiate them, and the shape of the graph changes radically. We lose the cells that have just replicated their DNA, we lose most of the cells with the normal DNA content, and we greatly enhance this apoptotic cell population. So all this says is that radiation causes apoptosis in these lymphocytes. That was no big deal, but it was important baseline. So now we look at the cells from the animal without P53. Here we are, without any radiation, and as we might expect, the cells are going around the cycle. They've got G1 content, G2 and S, and they've got a little bit of dying cells, just exactly the same as over here in the normal cell. But when you irradiate this population, I still get a thrill when I look at this graph, actually. It's exactly the same as it was before. These cells are dividing. They're going around their cell cycle, and yet they have received irradiation which knocks this normal cell to bits. But they are still going on in cycle. It is as if they did not hear, they did not attend to the fact that their DNA had been cleaved by the ionizing radiation. In fact, both these cell populations have a go at repair, and they do it together. So it's not the radiation that's killing the cell, it's not the fact that there's been damage and repair that's killing the cell, it's some other signal that says, I have been lethally injured. And if you don't have that signal, you don't die. So what do you turn into? You, in the sense of being a cell. Oh dear. So this actually summarizes a great deal of stuff. Here's a double-strand break, here's a normal cell, here's time, and if you've got P53, around the time when the radiation comes along and damages the DNA, then apoptosis is at least one of the endpoints. If you don't have p53, you end up with damaged DNA of a dramatic sort. We knew by this time that the cells, I call them the undead cells, because they're cells that should have died, but they have a DNA content which is bizarrely abnormal in terms of the way it's organized. And when you compare the sorts of changes that have happened in this little plastic experiment, the changes are not dissimilar to the changes that we see in authentic human tumours which don't have p53. This is the chromosome array in glorious technicolor, in a technique introduced by my predecessor here in Edinburgh, in Cambridge. It's historical, it's getting into my neurons. Martin Ferguson Smith produced this technology which allows you to colour the chromosomes individually in their own colour. And what you see here, this was a karyotype, a set of chromosomes from a cancer cell which had been originally derived from colon cancer. Now you know that there should only be 2 of each sort of chromosome. Well, there's 3 chromosomes only in this set which have just 2. These 2, and these 2, and these 2. And all the others, bizarrely abnormal numbers, and you can see from the colour that there's also been breaks in the chromosome, and then the chromosomes are stuck together. So there's been some sort of attempt at repair, but it's chaotic. And so this is the phenotype of the undead cell, the cell that could not turn on its death process. So this death process, you can see, is a preservative for the normal constitution, the normal life of the cell. And it's been recruited into DNA damage from a pre-existing developmental program. At least that's the interpretation that one could put on it. So now the change of gear. That's the biology, and that's basically how. The story's been modified quite a bit since these times, these heady days when we were sort of alone and you know, it was good fun. But I don't think there's anything in the necessary and inevitable revision period that has taken away the core of what I've just shared with you. But the question why, I don't think I'm able to answer why is there cell death, because there might have been other ways of putting tissues together that didn't involve it. But the question why that I would like to share with you and know your feelings about is why the scientific community changed so radically from being initially completely skeptical, almost completely skeptical. I suppose it wasn't really skepticism to appear in the Christmas issue of Lancet, but you know, it wasn't quite what one expected as a youngster. Why did the scientific community change from being essentially negative about cell death as a legitimate object for study to one in which there are now thousands upon thousands of articles on the process of cell death? What made the difference? Now, try to say this without arrogance, but I have 25,000 citations from my literature. It's a reasonably high number. Most of them are clustered around 3 or 4 publications, I have to say. What was it that suddenly took off this field? Why did people change in their view? Well, I'm going to offer you a few explanations. The first is that actually there were biological difficulties in identifying the dying cells. Here you see the difficulty. This is an irradiated bit of actually a colon cancer tumour, but you can see that there are some of these dying cells lying around, but they're surrounded and greatly outnumbered by something else, which is the normal cells. And the normal cells aren't quite normal. The normal cells are reacting to these dying cells. So, you can get terribly wrong with your biology if you just ground the whole thing up and look at it together. So, okay, it was difficult. to see. Secondly, although I made a bit of a song about it, despite the fact that the chromatin ladder was a pretty good and unusual signature, and once the caspase enzymes had been identified, there was a whole lot more molecules which indicated a cell in the process of cell death. Until these things came along, it was difficult to recognize cell death. People didn't like morphology as the only way of doing it. So it was difficult to see, it was difficult to recognize. Thirdly, it was difficult to fit in with existing evolutionary paradigms. Because how do you select for death and get anything at the end of it? At least that was the question that was raised. Actually, it's not too difficult. It is more difficult if you think of it as a single isolated cell, but most organisms are not single isolated cells. Even cultures of bacteria in real life are not single bacteria. They have a quorum sensing device which allows them to tell whether they're in company or not. As soon as you have two cells that are capable of talking together, the whole business of how you select for a death process which might preserve the life of a neighbour, then as soon as you get that, then the evolutionary argument is no problem at all. But there was some resistance on these three grounds. But I'm going to put to you the fact that I think there was something else as well. Just to dismiss the evolutionary argument, here is a little clip from Albert Weissman, was an evolutionary biologist who wrote prolifically at the end of the 19th century. There cannot, however, be the least doubt. Science was wonderful in these days, wasn't it? You could sit in an armchair and then come out with statements like this. There cannot, however, be the least doubt that the higher organisms, note the superiority, as they are now constructed, contain within themselves the germs of death. If you just replace the word genes for germs, he's bang up to date, actually, that's what I've been talking about. The question arises as to how this has come to pass, and I reply that death is to be looked upon as the occurrence that is advantageous to the species as a concession to the outer conditions of life, and not as an absolute necessity essentially inherent in life itself. It's quite a quotation, isn't it? So, I don't think it was the evolutionary argument that switched people off initially. I think it was this. This is the postmodernist view of life. What is my authority? Life in the USA, a complete guide to American life for immigrants and Americans. All materials on this site, copyright. Elliot Esmond, I had to include that. But look at this. Americans do not like to talk directly about death. In America, people don't die. There are various euphemisms. People about to die are terminally ill. Perhaps Americans have trouble talking about death because it is often so remote. People no longer die at home, but in nursing homes or hospitals. I'm interested in this next sentence because it is actually 2010. It's up to date, so I wonder how true it really is in America, but here we go. Insulated from disease by medical science and from the horrors of war by two great oceans, Americans have made death what sex once was, a subject only alluded to. This is the denial of death phenomenon. It has to be illness or weakness or violence. It can't be part of a program. And I believe that somewhere in the back of people's minds, why should they ever study death? Because it was just the end anyway. Well, this is where I think Christian apologetics have something to add to the debate. You don't get very far into the Bible before you meet with death presented in a very unusual context. so unusual that we, mostly speaking, miss it. Death is presented as a preservative. Man has become like one of us, said God, after the disobedience of the Garden of Eden. Knowing good and evil, he must not be allowed to reach out his hand and take also from the tree of life and eat and live forever. A premium issue in the mind of God as revealed in that scripture is that he will not have humanity enslaved in an eternity of sinnership. Remember, the context here is God searching for Adam. Where are you, Adam? And Adam and Eve hiding in the bushes because they had become aware of their nakedness. So yes, there was an element of penalty for sin, but it was also God's way of preventing that sin remaining forever in a living person. So, deliverance from permanent isolation. Reading on in the Old Testament, Exodus, Leviticus, bits of Numbers, Deuteronomy, full of a repeated message that the penalty for sin can be exacted legitimately, vicariously, by an innocent life. The sacrifices of the Old Testament, they were not pagan sacrifices. They may have seemed to be because they were contemporaneous with pagan sacrifices, but the motive in a pagan sacrifice is to placate an angry God by giving him a snack. Here, the motive is to find a means of approach to a holy God by the transfer of sinnership from the worshipper to the animal. And so we come to Christmas tide. And have you noticed, people who are working in Christian communities, and of course I don't know you, and I'm not making that assumption, but it seems to me either I've changed or the community is changing. You know, the hymns about the Virgin's womb and so on, I used to sing these as a boy without a thought. But every year now, I seem to hear this message. Have we really grasped what the humanity of Christ was all about? This scripture tells us he too shared in their humanity, so that by his death he might destroy him who holds the power of death. There was no way, even for God, to enter into the process of human death without becoming human. To free those who all their lives were held in slavery by their fear of death. So, Christ then appears in the New Testament as the vicarious sufferer on our behalf. And when that happens, a number of things happen together, but one of them is that the person who trusts that somehow is able to receive the indwelling presence of God. I couldn't think of an image This is, of course, Iceland. But I couldn't think of an image. You can see this thundering cascade coming down from above. That could not have been preservative were it not for the cleansing. So when we speak about the Holy Spirit indwelling people, we are serious as Christians. That can happen. Yes, we're still failing people. Yes, we still make mistakes. We park our cars in the wrong place. We offend our neighbours, all the rest of it. But nonetheless, God has come to dwell. And if the Spirit of Him who raised Jesus from the dead is living in you, He who raised Christ from the dead will give life to your mortal bodies through His Spirit who lives in you. I think it's terrific. And then we're very close to the end. Because death having been introduced as this preservative, as this stop signal, it stops the ongoing habit of sinful man, but it opens up a possibility of a new life altogether. And we call this new creation, we call it second birth, we have all sorts of names for it, but that's what the Scripture is talking about. The trumpet will sound. Death will be raised incorruptible, and we shall be changed. For this perishable must clothe itself with imperishability, and this mortal with immortality. And when the perishable has been clothed with imperishable, and the mortal with immortality, then the saying that is written will come true. Death has been swallowed up in victory. That's why I believe that it was easier for a Christian to understand what was going on in apoptosis than it might have been for somebody who had never met these ideas. I'd had this amazing privilege of growing up in a Christian family, so I had been exposed to this sort of thinking, that death was there as part of a program. It was part of a preservative program to ensure the normal health, the normal development of the person, of the created person. That was what I'd grown up with. Had I not grown up with it, every idea, every step into that logic would have been unfamiliar. As it was, I am still waiting for the trumpet to sound.
Great, thank you very much. Well, I'm sure that's stimulated all kinds of questions, biological and theological, no doubt. So over to you for question time. Who's going to be first? It's done. Peace, yes.
I was thinking as you were talking, when you first mentioned that it happens so that something else can happen. Somebody once said to me, we have to practice dying, we've just got to practice it, and when I put my ego aside, it might be in an infinitesimal situation in a very small way, but I have to accept that that's part of the, and I have to let that bit go so that I can live a bit more.
Yes.
And it just seems to me it's there all the time.
Yeah. Yes. Yes.
And I had never, because I hadn't heard all this, made a...
Yeah. I you know, it's an acutely personal thing for me, and I have some hesitation in sort of making a theology out of biology. But I think it was Kepler who used to write, you know, for the glory of God. When he saw a new nebula, or, you know, spotted something in the heavens, he saw the signature of God. And I think you can do that in our science. We can do that. But I don't know if I'm touching what your thought really was there. You were thinking when you said even if it's one little step. What were you?
In our living, in our moral living, our
Yes,
emotional
yes,
living, our
yes.
spiritual development, our psychic development, we have to let those moments that you've been describing to us happen
Yes.
on another plane.
Yes. Yes. Put to death, therefore. He says.
Peace, yes.
I think you painted a picture of Americans fearing death, or at least not talking about it.
Yeah.
And then you gave us a picture of Christianity being a good understanding of death, why it has a meaning. But we're also told that in America they've got the highest number of believers in the world. So how do those two things fit together?
Nobody can deny the facts, and nobody can, I think, come up with a really satisfactory explanation. I mean, you could even take it further, of course. You have a population which is replete with churchgoers who vote for invasion of foreign countries. Not easy. I think there is a theological explanation at a personal level, in that we often create a comfortable theology of our own without actually confronting the radical message of Jesus. That's about as far as I can go. I don't know, you may want to add to that.
Could I add just one recommendation of a book, Andrew? Andrew Putnam, the American sociologist, has recently published a book called American Grace, which is a huge tome on a contemporary analysis of the American religious situation. It's definitely the best recent analysis, in my view. I'm just in the middle of reading it, that there is around. So I would just recommend that as a very insightful, sharp sort of book on the sociology of American religion.
I think that, I mean, there are sociologists who have felt it's not just American, that we're all tainted. There was a chap, I didn't come across this name because I'm not actually much of a student of these things, Dennis, but there was a historian in France called Philippe Ariès, who wrote a lot about the changing cultural view of death, he said, across the Western world. I know he's been challenged because people said that you were French and you were talking about France. but he did, in fact, he outlines something very similar as a postmodernist denial of the existence of death. So this has been around for quite a long time. Whereas, you know, even outside of Christianity, there are heroic deeds done that, you know, the events of death can be conceived of in a kind of grand way, and I don't know where that fits into the whole picture. I've been interested to read the book.
More questions, please. Over to you. Yes.
Following on that question, can we push you a little further on the question that we hear about quite a lot these days, and that is termination of death prematurely? Induced apoptosis?
I knew somebody would ask that question. Of course, apoptosis is a cellular event, and it's going on normally. I mean, there are thousands of apoptotic events going on in this room right now. But I'm not certain that the analogy really holds firmly. But that I haven't lived with somebody suffering from motor neuron disease, so I can't enter into the emotions there. But I have read books of people, Joni Erickson being perhaps one of the more outstanding examples, with high spinal transection, whose life at one point seemed completely valueless, but in fact became of great value, obvious value to many people. So I guess I've sort of hinted at where I am in all of this. I would not feel at ease in a society which routinely practiced euthanasia. I'm getting fairly old myself.
Okay, other questions? Yes, please.
I must say the Christology of Hebrews is magnificent. What I'm anxious about is Paul's ability to grasp with infinity is always a bit wobbly. Now, we are finite, and as you have pointed out wonderfully, and as a complete non-science person, you've done me a great favour. I've actually learnt an awful lot. But what I'm anxious about is, as a biologist, tell us about infinity, because we're finite and we're dying. We're in the process of dying and living and being and so on. Tell me, as a biologist, about infinity. Because I feel it is there that we can actually say that Jesus Christ, and there is the paradox of Jesus Christ. The Christology of his humanity is actually magnificent. I really think if you would process that, it would do lots of theologians a lot of good to hear that, and a lot of people a lot of good to understand that his humanity is absolute, because he died in the process. But he is also God, the Son of God. So, as a biologist, how do you come to infinity? Or do you?
Well, you've given me the door out of it, actually. I know there'll be people in this audience who say, oh, he's copped out of it again.
No.
But I really don't believe that even if we used our tools in biology to their full extent and measured every gene, looked at the regulation of its transcription, looked at the functions of the proteins, and were able to put all these things together. And we're actually quite a long way away from doing that even now. But my belief is that even if we were able to do all of these things, that would not lead us to a perception of this immortal soul that we do believe we possess. Why do we dislike that idea? Well, partly because we can't see it and we can't measure it, and it doesn't fit in with our logic, which is where we were, and partly because we actually know there's nothing that we know that allows us to control that. And so it is a matter of faith. And then you're left measuring the value of that faith in human terms, which is some good, but will never make it cease to be faith.
So you're very happy as an eminent scientist to accept that we can't go to infinity. Infinity must come to the finite.
Yes. Yes. Yes.
But as a cell biologist, you can't build immortalized cells.
Oh yeah. Well, sort of. Yes. I mean, the whole risk could go. I forgot. Yes.
And your own stem
Yes.
cells.
Yes. That's right. But the interesting thing is that the stem cells are okay, Brian, for the life of experiments, but even transplanted stem cells, even if we could do that, there are problems down the road there. But the cells that really survive actually have genetic change in them. The germline. Yeah.
Okay, please, question here. Just think it.
You have to expand the story into cosmology, don't you, and into a wider area, because your immortalized cell lines are only immortal because you can put energy into them and feed them. So actually we're talking, when we think about infinity and other things, we're talking about the whole picture. And cell culture is, for some people, the whole picture, but for many people not.
Do we come back to the planet searching people? If we were on another planet, would we have these immortal cells?
Well, if you ask Simon Common Morris, I think he'd definitely say yes. I mean, there's no doubt about it. You know, that life anywhere is going to look boringly the same. So I think it's what many evolutionary biologists would say, but you know, we don't know.
I have this kind
Yeah.
of seeing. I live by cartoons partly, Dennis,
Mhm.
and I see all these cartoons showing the crashed flying saucer in the Arizona desert, and this little blob person moving along, and you can see the track had led out of the flying saucer, and he's saying, "Ammonia, ammonia, I want ammonia."
I think we talked to Simon, you know, he has sort of gin-sipping fellows on some far-off planet, you know, warming their backs against some fire in some far-off sort of Oxbridge College. I mean, you know, life is inevitable and will look very much the same, but I guess we have to wait for the data ready. But maybe Jennifer Wiseman will tell us on March the 1st. I think we do have time for one or two more questions, quick ones. Please, yes.
Isn't the thrust of what you've been saying that unless you have death absolutely central to the whole process of life right from the beginning, that if you thought you could get away from that, you wouldn't have perfection, you'd have a monster? And in a sense, that's what you're saying. Without that carefully regulated life and death, you end up with something which is like the cancer cell or whatever.
Yeah, I think so. I mean, I understand. I haven't studied this, but this is an answer, I think, to some of what Brian was throwing in, or perhaps you wanted to hear. Even unicellular organisms tire of replication. But you're absolutely right. The absence of death produces the abnormal.
Okay, one last question. Yes. Okay,
You have described the death of cells and the death of our cells and so on, but death surely is may be defined as the absence of life. Are we any closer to knowing what life, in fact, whether it's in a cell or a person, in fact exists, what it is?
I think biologically the answer is yes. I mean, there are people who would certainly claim to have put the bits together and given them a bit of a push, and oh, it worked. Yes? Whether we create life that way is a somewhat more tortuous thing to find one's way through. But I mean, I think you could create a set of statements through which you could test anything you picked up as the flying saucer man in the Arizona desert and say, is this alive or not? You could formulate questions which would allow you to answer that in terms of metabolism and sensory perception and so on. Some very basic things.
That is more the characteristics of a living organism, isn't it, than what actually life is?
That becomes a philosophical issue, absolutely. Yes, yes, yes.
Okay, well, we'll leave that hovering, I think, and we'll say it's time to stop there. But I think we've had a fascinating, a very provocative, stimulating, interesting seminar from Andrew. So let's thank Andrew very much.